to replicate is another word for copy. When we need to copy DNA, we need to be able to break it open first. All right, so the first step is to break it open, to break those hydrogen bonds. And the next step is going to be adding to those open ends bases, or actually nucleotides, that are complementary, A's with T's, C's and G's, to make a new strand on either side of the molecule of DNA. So that in the end, we're going to get two strands of DNA that are exactly alike. Part of it is going to be of the old molecule, and part of it is going to be a new strand of DNA, but these are going to be absolutely identical to one another. All right, so when we're talking about DNA replication, when we're talking about any process within the cell, we need to talk about the enzymes, the workers that are in the cell that actually do all the work, okay? So I'm going to show you first, okay, the 4 different enzymes that do work. This one over here in green unzips. This one over here is going to build by adding nucleotides, okay? This one right here is going to add a thing called a primer. It basically adds a starter. Just like you have to prime a wall before you paint it, that's a starter for your wall. This is going to be a starter for making a new strand of DNA. And then finally, we have our gluer together. Gluer, it's going to glue everything together, and that's really important to hold the new strand together. So let's just talk about all these again. So helicase is the one that we talked about first that does the unzipping. It unzips, okay? Next, we have DNA polymerase. DNA polymerase adds to make a new strand, okay? It's building. The next one is primase. Primase, okay, adds those primers. It adds the primer that you had to put on first before you actually can build or paint, right? Before you build a new strand. And then our last one is ligase. And ligase is the gluer. It glues everything together. So just to give you a little summary of this, again, we need to start with a double-stranded DNA molecule. We've got to separate the strands with helicase. We're going to need to make new strands, okay? And in the end, what we're going to get is we're going to have two identical strands of DNA that we made from the original strand. So thanks for listening, and I hope to see you soon.